Good afternoon. I am Phoebe Lewis, the Supplemental Instructor for Dr. Byron Quinn's Natural Science Biology 1 course at Langston University, and I have compiled a series of review videos that will help you, hopefully, with your final. This review video focuses on Chapter 3. Chapter 3 focuses on the pH scale. We're going to talk about acids versus bases, macromolecules and their monomers. We'll be covering carbohydrates, lipids, proteins, and nucleic acids, and briefly the functional groups. Now first let's talk about the pH scale. You guys remember that the pH scale ranges from 0 to 14, with 0 being towards the most acidic and 14 being towards the most basic. Now remember, acids are they release hydrogen ions into solutions, while bases accept hydrogen ions from solutions. Take a minute to just look at these examples. Know that battery acid, lemon juice, tomatoes, these are acidic examples of acids, while bleach and the lye that may go in your relaxers, ammonia, baking soda, are examples of bases. Now also remember that water and basically human blood are both relatively neutral with a pH of 7. Now let's talk about the classes of macromolecules. If you remember in class at the beginning of the semester, we talked about 4 main classes of macromolecules. We discussed carbohydrates, lipids, proteins, and nucleic acids. Now the building blocks or monomers of carbohydrates are your monosaccharides. Your building blocks of lipids are your triglycerides and your fatty acids. While the building blocks of proteins, you all should know this one, are your amino acids, and the building blocks of nucleic acids are, of course, your nucleotides. We're going to take a minute to discuss each one of these classes of macromolecules right now. First, let's talk about carbohydrates. Carbohydrates consist of long chains of carbon, oxygen, and hydrogen, and they consist of a huge part of most of you all's diet. They are made up of monosaccharides and disaccharides, which we consider simple sugars. Some examples of monosaccharides include glucose, galactose, and fructose, while disaccharides include maltose, lactose, and sucrose. Now, simple sugars can join together to form more complex sugars. We call this process dehydration synthesis or condensation. For example, monosaccharides can join together using dehydration synthesis to form disaccharides and polysaccharides, while the opposite of that, polysaccharides, can be broken down into their monosaccharide subunits during the process of hydrolysis. This figure right here, if we're going from left to right, from the reactant side to the product side, what you see is you have 2 monosaccharides, glucose. Two different glucose molecules are coming together, removing water and forming maltose. This is the process called dehydration synthesis or glucose. You can see why it's called dehydration synthesis, because you're dehydrating or removing a water. Now, the opposite of that, going from maltose, which is a disaccharide, if you want to get down to its subunits of glucose, you can add water to maltose and break that down into its subunits of 2 molecules of glucose using water, of course. That process is called hydrolysis. Now, different types of polysaccharides. Your polysaccharides are your complex sugars. You have starches, glycogen, cellulose, and chitin. Starches are commonly found in your potatoes. You guys are familiar with the starches. Glycogen, these are long chains of glucose. These are stored in your liver for energy. Cellulose, you guys know that you can find cellulose in the cell walls of plants, also in algae. And chitin is a common polysaccharide found in the exoskeletons of insects, including ticks. Lipids cover the second class of macromolecules that we'll discuss. Lipids are formed by triglycerides and fatty acids. So triglycerides and fatty acids are thus the monomers of lipids. Triglycerides are actually formed by a special type of reaction that we just discussed a minute ago. If you notice right here in the figure, you can see that the glycerol and the fatty acids are joined together by removing water and forming triglyceride. You guys remember what type of reaction this is? A condensation or dehydration synthesis. If you notice, the key is seeing that that water is removed. So dehydration synthesis occurs when glycerol and fatty acids join together to form a triglyceride. Now just to review, if a triglyceride is broken down into fatty acids and glycerol, how would we do that? Through a hydrolysis reaction. So breaking down is hydrolysis, putting together is condensation or dehydration synthesis. Just a quick review. Now, if you go grocery shopping, you'll see that there are several types of fats that are mentioned on the food labels of the groceries that you're looking to buy. You have trans fats and cis fats. Trans fats occur when the hydrogens are on opposite sides of the carbon chain, where cis fats occur when the hydrogens are on the same side of the carbon chain. Then you have the other infamous saturated versus unsaturated fats. Saturated fats occur where there are no carbon-carbon double bonds. So if you look here in figure A, this palmitic acid, you'll see that there are no carbon-carbon double bonds. All the carbons are thus completely saturated with hydrogens. Now if you look here, you see that this one has kind of a kink or bend to it. Well, that kink or bend is due to a carbon-carbon double bond, which means that this is actually an unsaturated fat. And so the unsaturated fats are considered unsaturated because there are actually double bonds. In this one, on figure C down here, linoleic acid, there are several double bonds, which means that this is a polyunsaturated fats. You can tell the difference just by if you're looking in your kitchen, you can tell the difference between a saturated and an unsaturated fat, because the unsaturated fats tend to be liquids at room temperature, where the saturated fats are solids at room temperature. So your Crisco shortening is going to be your saturated fat, whereas your vegetable oil that's liquid is going to be your unsaturated fat. So examples of lipids. We know that we have phospholipids that we discussed on the chapter covering the cell membrane. Phospholipids are the major component of the cell membrane. And we also have steroids, which are commonly found as natural hormones in your body, where you have testosterone and estrogen. These are actually examples of lipids. Cholesterol is another example of lipids. So you have testosterone, estrogen, cholesterol, and phospholipids that are all examples of lipids. So let's discuss real quick. We talked about so far carbohydrates and lipids. Well, what are the differences? If you notice, carbohydrates and lipids are both made up of the same elements: carbon, oxygen, and hydrogen. Well, if they're both made up of these three elements, how can we tell the difference? Well, the major difference is that carbohydrates are soluble in water. So your examples, your sugars. You know that if you pour some sugar in water, you can mix that up and it will dissolve. Well, lipids, on the other hand, are insoluble in water. You always know, you've been told since you were a young child, that oil and water don't mix. And that is, of course, true in this case. So that is one way to know the difference between carbohydrates and lipids. Well, the third class of macromolecules that we'll be covering is the proteins. Proteins are made up of amino acids. So the monomers of proteins are, of course, amino acids. Amino acids all have this amino group, a carboxyl group, and then a side chain, which is it can be any type of chain of carbons. Well, what makes the amino acids different from each other are, of course, the side chains. When amino acids link together via the infamous condensation or dehydration synthesis reactions, they form polypeptides. So polypeptides are just long chains of amino acids. Proteins actually have several levels of structure. You have the primary structure, which is the most simple structure, and that is just simply the chain of amino acids. Then you can have these chains of amino acids will actually fold up into different structures, including the beta pleated sheet, the alpha helix sheet, or the random coil. And when they fold up into these different shapes, you have the secondary structure of protein organization. Then, when these structures join together, you have different of the secondary structures coming together. You form the tertiary structure, which is simply a folded polypeptide chain. And then the quaternary structure occurs when you have more than one polypeptide. And so 2 or more polypeptide chains form the quaternary structure of protein organization. The last class of macromolecules that we'll discuss today is the nucleic acids. Nucleic acids make up the DNA and the RNA. You guys have heard these before: deoxyribonucleic acid and ribonucleic acid. Nucleic acids are made up of nucleotides. So nucleotides are thus the monomers of nucleic acid. Now nucleotides are interesting because they actually have some subunits to them as well. So nucleotides are made up of, you guys have learned it, a phosphate, a sugar, and a nitrogenous base. So each nucleotide is made up of a phosphate, a sugar, and a nitrogenous base. So in the case of DNA, you have a phosphate, you have the sugar deoxyribose, and you have one of 4 nitrogenous bases, either cytosine, guanine, adenine, or thymine. And we'll discuss those later on in later chapters, but that is all you need to know so far. Alright, just to review, we covered, you notice that in the macromolecules, the structural backbones of these macromolecules are your functional groups. Your functional groups are your carboxyl, your hydroxyl, your amino, and your phosphate groups. The phosphate group we already discussed in the case of DNA, and you'll learn in the next couple chapters about ATP, but phosphate groups make up one of the functional groups. They're seen in your nucleic acids. Amino groups are seen in your amino acids, which make up what? Proteins. Okay? Hydroxyls, the hydroxyl group are seen in your alcohols and your carbohydrates. What are the monomers of carbohydrates, just for a review? The monomers of carbohydrates are, of course, your monosaccharides. Your carboxyl groups are found in your fatty acids and your amino acids. What do amino acids make up? The answer should be proteins. What do fatty acids make up? Lipids. Okay? And what are the monomers of DNA or nucleic acids? Nucleotides. Okay, so let's just do a quick review. If the monomer is monosaccharides, the polymer is polysaccharides, and these are your carbohydrates. If the monomers are your amino acids, your polymers are your polypeptides or your proteins. If your monomers of your are your nucleotides, your polymer are your nucleic acids. And then if the monomers are glycerol and fatty acids, the polymer is a lipid. Alright, and that covers chapter 3.